Welcome to the CRISPR-Cas video series of the project Genetic Engineering and the Environment. The topic of this video are the possibilities of CRISPR-Cas. My name is Katharina Kawal. I have a doctoral degree in molecular biology and I will guide you through this video. The previous video described how CRISPR-Cas works and ended with an overview of possible alterations that can be induced in the genome by using the gene scissors. I would like to go through this overview again as it is important to understand this video. With the simplest CRISPR-Cas application called SDN1, in which only small changes in the genome are induced, genes can be knocked out or their functions can be altered. Furthermore, genes can be deleted from the genome or the gene expression can be altered. In this video, I will show you that a combination of these possibilities enables multiple and complex changes in the genome, which can result in the generation of new genotypes. A genotype is an organism's complete set of genetic material. I will explain step by step how the complex changes to DNA are made using only the simplest type of CRISPR-Cas and how new genotypes can be generated. CRISPR-Cas enables the alteration of several identical DNA sequences, as well as the alteration of several non-identical DNA sequences, which is also called multiplexing. In addition, linked genes and protected parts of the genome can be altered. Firstly, I'm going to talk about the alteration of identical DNA sequences. Sometimes the impression is given that using SDN1 applications of CRISPR-Cas, only a single point mutation in the genome is induced. However, this is a gross oversimplification. With CRISPR-Cas, all DNA regions with the same target sequence can be altered. Plants often have multiple copies of the same chromosome in their genomes, such as hexaploid wheat with a 6-fold set of chromosomes. So there are 6 copies of a single gene. If a guide RNA, which the gene scissors use to identify the target sequence in the genome, is introduced into the cell together with the gene scissors, the target sequence of the DNA cannot only be recognized and cut once, but rather several times. In the end, up to 6 changes can be made at once. So all DNA regions with the same target sequence can be altered. Using one single guide RNA, alterations of several identical genes in polyploid plants, meaning plants with more than 2 sets of chromosomes, can be induced at the same time. In addition, multiple copies of a gene can be changed at once. This is particularly relevant for alterations or knockouts of genes in gene families. Gene families arise when genes duplicate multiple times. Gene families can contain several hundred genes and can be present in one part of the genome or distributed within the genome. Gene families are common in plants and play an important role in the development of new plant species. I have 2 examples from the scientific literature for you that show what can be achieved by altering several identical DNA sequences. In a wheat, for example, 35 of 45 alpha-gliadin genes were altered at the same time by using CRISPR-Cas. Alpha-gliadins belong to a large gene family and are the main component of gluten, so the wheat contains less gluten. Another example is the cultivated strawberry, which has an 8-fold set of chromosomes. In this study, multiple copies of a particular gene were altered at the same time, resulting in an altered development of the anthers that produce pollen. These 2 examples show that even the simplest type of CRISPR-Cas can cause profound changes to the plant by altering multiple copies of a gene. Next, I would like to explain to you a genome editing approach called multiplexing, which means changing several non-identical DNA sequences using different guide RNAs. In multiplexing, several guide RNAs are introduced into the plant cells together with the gene scissors, indicated by the different colors of the scissors. This means that several different DNA regions are changed at the same time. Of course, multiplexing doesn't just change individual copies of the target genes, but rather several identical DNA sequences are changed, which is why multiplexing occurs in combination with changing identical DNA sequences. If we look at the example of wheat with its 6 chromosome sets, it means that if 3 different guide RNAs are used, up to 18 DNA sequences can be changed at once. The question arises as to what is the purpose of changing several genes in a plant at the same time. Well, many traits in plants, for example their stress response, depend on several different genes, and alteration of such traits is considered best achieved with the multiplexing approach. It is questionable whether these plants can prove themselves in cultivation. Furthermore, the complexity of the possible applications of the gene scissors increases the probability of unwanted changes in metabolic pathways. For example, if a gene is knocked out that is involved in several processes in the cell and whose functions are not yet fully understood. I go into more detail about such unwanted effects on natural interactions in the video about the risks of CRISPR-Cas. I have also summarized a few examples for you that show how complex the applications of CRISPR-Cas can be. In another application, in wheat, 3 different genes were altered to increase the yield. In a rice, 8 different genes were edited or altered to induce a higher yield, a different growth pattern, and a different fragrance. These and other examples show how potent applications of CRISPR-Cas can be. CRISPR-Cas can also be used to alter so-called linked genes. Genes that are very close to each other on a chromosome are referred to as linked because they are mostly, meaning very likely, inherited together. If 2 genes are located on different chromosomes or on the same chromosome but with a greater distance between them, then they are not linked and can be inherited separately from one another. With CRISPR-Cas, linked genes can be separated, for example by knocking out one of the 2 genes via the induction of small changes in their DNA sequence, making them non-functioning. This can be used to interrupt the common inheritance of certain traits. I also have 2 examples from the literature for you. One is about a genome-edited tomato in which 2 genes are separated from each other, which determine the shape of the fruit on the one hand and the connection between the fruit and the stem on the other. With CRISPR-Cas, the joint inheritance of these 2 traits was abolished, so that the tomato has a specific fruit shape and is easier to harvest. In addition, alterations can be introduced directly into elite lines, without the backcrossing process through which one normally segregates genetically linked genes. With CRISPR-Cas, certain traits can be generated directly in elite lines, such as this genome-edited corn here, which tastes sweeter and has a higher starch content without backcrossing. We are coming now to the last point of our list, the alterations of protected parts of the genome. The classical theory of evolution says that in nature, new mutations can occur in all genes with the same frequency, regardless of the gene function and the consequences for the fitness of the organism. However, a number of scientific papers have been published in recent years which show that new mutations naturally occur more frequently in certain areas of the genome than in others. This is shown by work with Arabidopsis thaliana, a model plant used in basic research, but also by work with human cells and yeast cells. Mutations are therefore not evenly distributed in the genome. Fewer mutations occur in many functionally important areas of the genome, such as in gene areas. The mutation rate depends on certain repair mechanisms that are responsible for repairing newly occurring mutations in such regions. The location of a region within a gene also plays a role. It has been shown that more mutations occur in the outer parts of genes than in the inner parts, which often control the gene's function. It was also shown that specific epigenetic markers are enriched in the gene regions that are particularly protected by the DNA repair mechanisms just mentioned. Epigenetic markers are inheritable factors that do not alter the DNA sequence and regulate gene activity. You can think of these epigenetic markers like switches that are enriched at genes and turn them on or off. Using CRISPR-Cas, alterations can be induced anywhere in the genome. This results from the mode of action of the gene scissors. In case a DNA double-strand break is induced by CRISPR-Cas and restores the original state, the gene scissors can recognize the target sequence again and cut there. Ultimately, CRISPR-Cas increases the possibility that alterations can occur in such protected regions of the genome. Using CRISPR-Cas, it is possible to change gene regions in which mutations are naturally very rare. I also have an example for you. In this study, a specific region of the target genes was altered with CRISPR-Cas in such a way that the fruits of the tomatoes contain more GABA, a neurotransmitter, which is supposed to have an antihypertensive effect. Chemical mutagenesis has not yet been able to introduce a mutation in the specific target region, whereas CRISPR-Cas has. I have shown you in this video how the application of the simplest CRISPR-Cas application, SDN1, can generate complex alterations in the genome of plants. Complex alterations result from several small changes to the DNA sequence. As a result, plants with new genotypes can be developed. In the next videos, I will go into more detail on the risks of CRISPR-Cas. I will talk in one video about the unwanted effects such complex changes can have on natural interactions. In another video, I explain to you which unwanted changes in the genome can be caused by the use of the gene scissors and the use of old genetic engineering methods. Thank you for your attention. You can find more information at the website of the project Genetic Engineering and the Environment.